Hello and welcome back to another exciting edition of Ed Puzzle Notes. Today we're going to continue in our journey through genetics. We're going to learn how to use Punnett Squares to determine probabilities of phenotypes or traits in the offspring. But first we're going to do a little review on these very key terms over here. So a lot of these terms you've probably never seen before. They're totally new. Don't worry, if you practice them, you will definitely learn them. But on the other side, if you don't practice them, you probably won't. Now, first term is a gene. A gene is a segment or section of DNA which codes for a particular trait or feature or characteristic that you have. Now remember, we have 23 pairs of chromosomes. So we have 2 of each chromosome. We get 1 from mom, 1 from dad. So since we have 2 of each chromosome, we also have 2 of each gene. So we get 1 gene from mom, 1 gene from dad. Now, we all have hands and fingers. But how does our DNA tell our body how many fingers we're supposed to have? Well, we all have a gene for this. That particular gene is located on chromosome pair number 7. Now, for those of us that have 5 fingers, like this person over here, 1, 2, 3, 4, 5 fingers, that is a specific allele. So an allele is the different types of genes. Now, the 5-fingered allele is what's called recessive. That means the only way you can have 5 fingers is you have to have 2 recessive alleles. And we show recessive alleles or recessive traits with lowercase letters. We'll use an F for fingers. Now, over here, you can see this person actually has 6 fingers. That is a different allele. This person has the dominant allele, at least one. And we'll signify that with a capital F. So, lowercase f, or lowercase letters, those are recessive, and you got to have 2 of them. So the only way you can have 5 fingers is you had to give a recessive allele from your mom and a recessive allele from your dad. Now this, when both alleles are the same, either they're both big or they're both little, that's called being homozygous for a trait. The prefix homo means same. So if you have 2 of the same alleles, you're homozygous for that trait. And since they're both so small, lowercase letters, we're going to say you're homozygous recessive. Now, for the people with 6 fingers, I've never met anybody with 6 fingers, but it does happen. They can have 2 different what's called genotypes. You can have 2 different genotypes if you have 6 fingers. You can only have 1 genotype if you have 5. You got to be little f, little f. Little f genotype gives you the phenotype of having 5 fingers. But people with 6, they can have 2 different genotypes. They can be big F, so they got 2 dominant alleles. That's homozygous again because they're the same. So they're homozygous dominant this time. So they got a big capital F from their mom and a big capital F from their dad. Gives them 6 fingers. Now, the other possibility is you could have capital F, dominant allele, and 1 lowercase allele. So they got a capital dominant allele from one of their parents, and they got a lowercase recessive allele from the other parent. Whenever you have 2 different alleles, a recessive and a dominant, that's called being heterozygous for the trait, or they also call it being hybrid. So heterozygous and hybrid mean the same thing. Now moving on to the pictures on the bottom. This little dent on that girl's face, that's called a dimple. Dimples are dominant traits. So we will signify that with a big D for dimple. Now this guy over here has no dimples. And we will signify that with a lowercase d. So, the only way you cannot have dimples, you've got to be what's called homozygous recessive. You got to have 2 little d's in order to not have dimples. That would be your genotype. So the only genotype you can have to not have dimples is homozygous recessive or little d, little d. Now remember, those of us that do have dimples, not me, but those of you guys, if you do have dimples, you can have two different types of genotypes. You could be big D, big D. So you got the dimpled allele from both parents, or you could be big D, little d. So you got one dimpled allele, the dominant allele from one of the parents, right there, and you got the recessive no dimple allele from your other parent. But it doesn't matter, since it's dominant. Whenever you have, it doesn't matter if you have two dominant alleles or just one. These two right here will have the same phenotype of having dimples. So it doesn't matter if you have two big Ds. That doesn't mean like you have more dimples or your dimples are deeper. No, they're the same. You can have two dominant alleles or just one. Doesn't matter. You still have the same phenotype of having dimples. All right, on to the Punnett squares. So Punnett squares, you use them to help determine the probability of different genotypes and phenotypes in the offspring of two parent organisms. So probability, what is probability? These are obviously quarters, and I'm guessing you've probably flipped a quarter to see if it's heads or tails. Well, the probability of that is 1/2, or 50%. It's a 50% chance of flipping a heads or flipping a tails. But what if you flip two coins at once? Now what are the chances of both of them being heads, or both of them being tails, or having one with the heads and one with the tails? What are those probabilities? So remember, in genetics, when we make our gametes, we only give one of our chromosomes to our gamete. We don't give both. We don't give both pairs. We just give one of the two. So this Punnett square is going to help to determine what's the probability of giving different traits in your kids, like dimples or blue eyes or 6 fingers or whatever. So this is how it works. Whenever you flip a coin, you know there's only two outcomes that can happen. You can get a heads or you can get a tails. So, same thing with our genes, our alleles. There's only two options. We can give the one from our mom that we got from our mom, or we can give the one we got from our dad. So, if we flip a coin, we can get a heads or we can get a tails. When we flip the second coin, it's the same options. You can get heads or you can get tails. It's not going to land on its side and stay balanced and not be neither head or tails. It's going to be one of those. So with these Punnett squares, all you do is you fill in the boxes. First, you put the two possible outcomes on the sides, and then you just fill in the boxes. So this box would be H, 2 heads. This box over here, you bring the H this way, and the T comes down, so it'd be H, T, heads and tails. Same thing with this box. We'll bring the heads from way up there, and the T's right there, heads and tails. And this very bottom right box, the T comes all the way down, and this T comes across. It's going to be tails, tails. Now all you do is you count them up. What are the chances of both quarters being heads? Well, there's one that's head, and that's it. 1 out of 4 chance you will get both of them heads. What are the chances of getting heads, 1 heads and 1 tails? Well, there's one right there, and there's one right there. So that is 2 out of 4, or 1/2, or 50%, however you want to say it, means the same thing, are going to be 1 heads, 1 tails. And last, what are the chances of just getting 2 of them with tails? Well, there's only 1 out of 4 boxes has tails, tails. So that goes back to 1 out of 4 chance of getting a tails, and the other quarter being a tails. So that's how it works. But remember, we're not going to be using coin flips. We're going to be using genes or alleles. Okay, now we'll try one for real. So here we're going to cross 2 little rabbits. A black rabbit with a white rabbit. Now, remember, both these guys have 2 genes for these 2 alleles for their traits, for their color. And they got it from their parent rabbits. You know, their parents gave them their black fur or their white fur. Now in this case, the black fur is dominant, big B. Lowercase b is for white. It is recessive. So we should know the genotype of the white rabbit right off the bat. Because the only way you can be a white rabbit is you got to have two recessive alleles, little b, little b. Now we're going to cross that with a homozygous dominant black rabbit. Homozygous tells us that both letters are the same, and since it's black, we know it's going to be, they're both going to be big. So we're going to cross big B, cross with a homozygous recessive or little b, little b. Now all you do is, so that's step one, figure out the genotype of the parents. So we got that. Step two is just filling out the box. So you're going to put these alleles, doesn't matter if you put them on the top or on the sides, you just got to make sure you keep them together. The big B, I'm going to bring them over here and put them on this side. So we're going to put big B. That is this guy's two possible alleles. Now he has the same, so all his alleles are the same. They're both big B. Same thing with the white guy up here. We're going to put its alleles right here. Little b. And then it's just filling in the boxes. Capital B, lowercase. I always say big B, little b, big B, little b, big B, little b. Genotypic ratio. That's just the ratio of all the different genotypes that we get. Now, if you notice, they're all the same. All four boxes, 1, 2, 3, and 4, have the same genotype of big B, little b. So our genotypic ratio, 4 out of 4, or 100%, are going to have the genotype of big B, little b. Now, phenotypic ratios. Basically, that's saying, what are these rabbits going to look like? Well, they're all heterozygous. They all have one big letter and a little letter. But since they all have the dominant black allele, all these little bunnies, 4 out of 4, and it doesn't mean they have 4, but 100% of them, so 4 out of 4 or 100%, are all going to be black. So all the rabbits, all the baby rabbits, 100% of them, are going to look like this guy. So the one dominant allele, black crossed with a white, doesn't give you gray. It gives you black because the black allele is dominant. Now we're going to try crossing two of the babies that we made on the last slide. So two of those babies, we're going to cross two of those guys. So they're both black, but if you remember, all the babies in the last slide were heterozygous. So that's step one. Figure out the genotype. Put heterozygous into two letters. Remember, we're using the letters B in this case. And since they're heterozygous, hetero means different. So both these rabbits are going to be big B, little b, crossed with a big B, little b. Now, it doesn't matter which of these two guys, if you put their alleles on the top or the sides, it doesn't matter. Just make sure they stay together. So I'm going to put this guy's up here. They're the same, so it really doesn't matter. So we're going to put big B, little b. And I'll put this guy's alleles over here on the side. Big B, little b. Now it's just filling in the boxes. This box right here is going to be big B, big B. This one over here on the right is going to be big B, little b. On the bottom left, it's going to be big B from up top and little b from the side. And on the bottom right is going to be little b from the left and the little b from the top. Now, the genotypic ratios, it's just about counting them. How many of them are big B, big B? I see one, I see one. 1 out of 4 will have the genotype of big B, or homozygous dominant. How many of them are big B, little b? I see 1, I see 2. 2 out of 4, or 1/2, are going to be heterozygous, or big B, little b. And the last box we have right here, 1 out of 4 of the boxes are going to be the genotype little b, or homozygous recessive. Now the phenotypic ratio, it's basically saying, what are these 4 rabbits going to look like? What are the chances of getting black or white? All right, so which ones are going to look black? This one's going to be black, that one's going to be black, and this one's going to be black, because they all have at least one dominant allele. Remember, you only need one to have black fur. So I see 1, 2, 3. So that would be 3 out of 4, or 75%. 75%, because 3 out of 4 is 75%, are going to be black rabbits. So that leaves us this little box over here on the bottom right, little b. So that would be 1 out of 4, or 25%, are going to be white. Now remember, this is just on average. Doesn't mean if these two guys had 4 little bunnies, they could all be white. The odds are against it, but they could all be black, they could all be white, it could be 50-50. But on average, 75% will be black, 25% will be white. Now I know what you guys are saying. You guys are all saying, Aroso, this looks fun. Can we try one? Well, the answer to that is yes. Let's try this one. This one's for you to try. Remember, step 1, turn these words into genotypes. So turn heterozygous tall into 2 letters and turn short plant into 2 letters. And we're using the letters T. Capital T equals tall. That's the tall allele. Lowercase t equals the short allele. These are plants. So see if you can do that. All right, well, hopefully you're able to do it. I will find out when I see your results. But this is how you do it. Step 1, heterozygous tall. I'm going to turn that into 2 letters, into 2 T's. Hetero means different, so I know I'm going to have 2 different T's here. So it's going to be a big T and a little t. And we're going to cross that with a short plant. Now, since short is recessive, I know the only way you can have a recessive or a short plant is they got to have 2 little t's, 2 recessive alleles. So there we go. We have the 2 genotypes of the parents. Big T, crossed with a little t. Now, doesn't matter which of these plants you put on the top and which you put on the bottom, but I'm gonna put the little t on the top. So I'm gonna go little t, and I'm gonna put this heterozygous alleles on the sides. So I'm gonna have big T, little t. And now it's just filling in the squares. This one's gonna be big T from the left, little t from up top. This one's gonna be big T from the left again, little t from up top. This one's going to be little t, and this one's little t from the left, and little t from the right. Genotypic ratio, that's just about counting them. How many of them are heterozygous, or big little? I see 1, I see 2. So there it is. 2 out of 4, or 1/2, or 50%, whatever you like to say, are going to have the genotype of big T, little t. And now the other one. I see 2 out of 4 are little t, and 2 out of 4 are little t. So 2 out of 4 are going to be homozygous recessive, little t. Now, what are these plants going to look like? Well, that's easy. 2 out of 4, these 2 up here, both have at least, well, they have exactly 1 tall allele. Therefore, 2 out of 4 are 1/2, or 50%. I'll put 50% this time. 50% of the plants, on average, are going to be tall because they have 1 tall allele. Now the other ones, these 2 down here, 2 out of 4, or 50%, are going to be short. So 50% are going to be tall, and 50% of the plants will be short. And now one more for you to try. Hopefully you're getting the hang of it. So remember, this time we're doing flowers. So we're going to use P. Purple is dominant. Big P is for purple. Little p is for white flowers. So step 1, turn heterozygous purple into 2 letters and turn hybrid purple into 2 more letters. See what you get, fill in the boxes, and we'll see how you do. All right, so heterozygous. Heterozygous means different. So over here we're going to have big P, and to make it different, we're going to have a little p. And we're going to cross that with hybrid purple. Remember, hybrid and heterozygous mean the same thing. Hybrid means different again. So we're going to have another one that's going to be big P and a little p. All right, and then it's just put the alleles on the top and on the side. So on the top here, we're going to have a big P and a little p. On the side, we're going to have a big P and a little p. Then fill in the boxes. This one's going to be big, big. This one's going to be big from the left, little guy from the top. This guy's going to be big from the top, little guy from the left. And this guy's going to be little, little. Now the same thing like usual. Genotypic ratio. How many of the 4 are big P, little p? Or actually big P, homozygous dominant? I see 1 out of 4. So 1 out of 4 will have the genotype of big P, or homozygous dominant. I see 1, 2 that have a big P and a little p. So 2 out of 4, or 1/2, or 50%, I'll just put 2 out of 4, are going to have the genotype big P, little p. And don't forget about the last one. 1 out of 4 will have the genotype of little p, or homozygous recessive. So 1 out of 4 are going to be little, little. Phenotypic ratios. This one has 2 dominant alleles. It's definitely going to be purple. This one has 1 dominant allele. That's all you need. 1 is just as good as 2. And same thing with this one. So that plant, and that plant are all going to have purple flowers. So 3 out of 4, or 75%, are going to have purple flowers. And the last one, this guy down here, little p, homozygous recessive. On average, 1 out of 4 of these flowers will be, these plants will have white flowers. All right. Thank you very much for watching. I hope you understand, and I'll talk to you next time.